next topic is going to be action potentials, excuse me, and saltatory conduction. So I put a little graphic up there. I'll have a couple of more on the screen. Let's see. We'll talk about saltatory conduction in a couple of slides, but I definitely think it's worth talking about the resting membrane potential because it does come up on the MCAT pretty often in my experience. So the resting membrane potential, that should say, for a human neuron is about -70 millivolts. So what that means is that when you're looking at the inside of a cell and the outside of a cell across the membrane, the difference between the two charges on the inside and the outside is 70 millivolts. So the convention is that whatever that number is, you're talking about the inside of the cell. So if it's -70 millivolts, that means that the inside of the cell is 70 millivolts more negative than the outside, less positive than the outside. And how does the cell maintain this? How does the cell keep charge? Through ions. So there are two pretty important proteins that you should know about. It's maintained by the sodium-potassium ATPase. It's also called the sodium-potassium pump. So it pumps 3 sodium ions out of the cell and 2 potassium ions into the cell for every ATP used. So this is an active process, as I'm sure you know, because we're pumping these ions against their concentration gradient, their electrochemical gradient, I should say. So sodium, there's a high concentration outside of the cell, a low concentration inside. So if we want to pump it from in to out, we have to use ATP. And it's the same thing with potassium. Potassium concentration is high inside the cell, it's low out. So it's an active process when we pump potassium. So the other protein is a potassium leak channel. These don't use ATP, it's passive diffusion. So what the leak channel is, it's basically just an open channel that allows the cell to be slightly more permeable to potassium. So it's just passive diffusion. Potassium is going to flow down its electrochemical gradient. And as we just said, since potassium concentration in a normal cell is higher on the inside, the potassium is going to flow out. So the two of these work together and they keep an equilibrium. So resting membrane potential for a polarized human neuron is -70 millivolts. So the neuron at rest is polarized. When the dendrites receive a signal, the membrane starts depolarizing. So depolarizing is always describing the first half of the action potential. We are at -70, which is a pretty polar value. It's kind of far from 0. So as it approaches 0, it is depolarizing, hence the name depolarizing. It also does pass 0 and overshoot. It goes, I think, to about 35 or 40. Opal said, what concentration is high where? So sodium concentration on resting neuron cells are higher outside of the cell. Sodium is always outside of the cell. Potassium is always high inside of the cell. Those are two things that you should definitely commit to memory because it comes up in, sometimes it'll come up in other topics other than the nervous system. Was that your question, Opal?
Yes, it was. Thank you.
Great, no problem. Okay. Yeah, definitely. I can repeat that. So sodium ions are higher. They have a higher concentration on the outside of the cell, and potassium ions have a higher concentration on the inside of the cell when it's just a neuron that's at rest. And then the sodium-potassium pump and the potassium leak channels, they work constitutively to keep this equilibrium. Yes, and Janelle put that in the chat so you guys can see it as well when I move to the next slide. Okay. So now we can get to the depolarization. So I put another graph of the action potential right there. At this point, I'm sure we've all seen this graph a lot. So input received by the dendrites initiates a slight repolarization. Summation, I also wanted to mention really quickly. It's the process by which a neuron adds all excitatory and inhibitory input to determine if the action potential threshold will be reached. So what I mean by that is that if you have a single nerve that's receiving input, most of the time there are going to be multiple sources for that input. So there could be hundreds of neurons that are leading up to this. So all that's saying is that it's going to add all of the excitatory or inhibitory input from all of those neurons. And if it reaches that threshold of 55 that you see right over there, then the action potential is triggered and it's an all or nothing response. If you look a little bit more to the right where you see those failed initiations, sometimes the neuron will receive input, but it's not enough to raise that potential. It's not enough to raise the resting membrane potential past the threshold. So the failed initiation, nothing happens, and then the sodium potassium pump and the potassium leak channels just gradually bring it back to equilibrium right by that -70 value. So not all input turns into an action potential, but any input that reaches the threshold is a 100% all or nothing reaction. So, like I just said, if the membrane potential reaches the 50-55 millivolt threshold, the cell undergoes complete depolarization. The subsequent rapid depolarization is due to the opening of voltage-gated sodium channels. So that's what happens that's why the -55 millivolts is significant, because at that point, that is the proper voltage level that the voltage-gated sodium ion channels open. So sodium ions flow into the cell down their gradient, and it increases the charge inside the neuron. So take a minute and kind of like intuit that, wrap your head around it, because it makes sense when you think about it logically and think about it while you're looking at the graph. So once you reach the -55, before the -55, it's just these little depolarizations that were started by the input. So once it reaches that 55, all of the cells' sodium gates swing open really fast, and there's a huge influx of positive ions. So we're at -70, and we're getting a huge influx of positive ions. So that's going to rapidly go up past 0 up to about 40. So the depolarization propagates as an impulse down the axon to the synaptic terminals, where it's converted into a chemical signal, and it's released into the synaptic cleft as neurotransmitters. So that's when the electrical signal is converted into chemical. And that's how neurons synapse, and that's how they interact with each other and communicate with each other. So what channels open during each stage? So for this stage, you just have to know, we're going to go over, I think in the next slide or two, we're also going to go over how the voltage-gated potassium channels work as well. But those are more important for the repolarization phase after the peak. So yeah, so they are important, but for this specific section, just know that once the threshold is hit, the sodium channels swing open, and they swing open very rapidly. So that causes a rapid influx of positive ions, and that's what triggers the action potential. And once they're opened, you can't close them unless we go through a process that I'm about to talk about.